My name is Emma, and I'm a summer REU student at Harvard's School of Engineering and Applied Sciences. I go to Northwestern, where I'm studying biology and biotechnology, and my main goal in learning to conduct scientific research is to save patient lives. I hope to one day be able to apply my knowledge and my skills to help develop medicines and medical technologies that will be able to cure patients all over the world. This summer, I'm working on a collaborative project between the Eisenberg Lab in the engineering school and the Mazur Lab in the biophysics department to develop a new kind of cell therapy. Cell therapy involves taking sick cells out of the patient's body, treating them in the lab, and then injecting or infusing them back into the patient where they ideally return to their native environment. And this approach basically lets us treat the patient from the inside out, which is pretty cool. So oftentimes, treating the cells in the lab involves delivering drugs or medicinal molecules past the cell membrane and into the cell. But this in itself is quite a challenge simply because the cell membrane is very picky about what it does and doesn't let into the cell. There are many chemicals and viruses that are commonly used as drug delivery agents, but problems do arise because these methods aren't compatible with all cell types and can have unwanted side effects like activating an immune response or simply being toxic to the cells. And regarding cell therapies, there are some cell therapies currently in use, but it's still very difficult with any existing method to produce high quality, viable, therapeutic grade cells in large quantities. So that's what our approach hopes to fix. So our approach uses microfluidic devices, or teeny tiny tubes shaped almost like silly straws, but with a dimension of a single cell, paired with a laser to physically induce temporary pores into the cell membrane through which our molecular cargo of choice can then enter and ideally begin its therapeutic effects on the cell. And this membrane has many pluses. It can go through many cells at a time. It can be used regardless of specific cell type because it's physical, not chemical. And it doesn't have any long-lasting harmful effects on the cell because the pores are temporary and close soon after they are formed. I've definitely learned a lot this summer, both in terms of theoretical scientific knowledge and hands-on lab techniques. But one theme I'm really starting to internalize is that research is just one big adventure, and that comes with many unforeseen obstacles that must be overcome through failure first, and creativity, and a lot of persistence, and without which the final result could never be achieved. This project has also showed me how traditionally different skill sets can actually complement one another and provide new insights on different aspects of the project. So far this summer, I've engineered experimental setups, fabricated microfluidic devices, tried to get them to stop leaking, operated a class 4 laser safely, frozen cells in liquid nitrogen, and among other things, used a scanning electron microscope to image our cells so we can visualize them and better understand the results that we're seeing. This has been my first time using a microscope of this caliber, and it has honestly been mind-blowing. And all of this while simultaneously maintaining cell cultures and evaluating which chemicals we can safely use with them to achieve our project goals. And the reason I'm able to do all this, I think, stems in part from the interdisciplinary nature of Harvard's engineering school and its direct affiliations to other Harvard schools. I'm truly grateful to have had this opportunity at Harvard and look forward to learning and growing as a researcher so I could one day contribute to the development of next generation therapeutics.